Where else would you rather be than a Zoom full of Penn Staters? I'm Paul Clifford, CEO of the Penn State Alumni Association, and I'd like to welcome everybody to today's virtual speaker session titled Peeking into Peptides, Decoding the Brain's Messages and Manipulating the Molecular Mailmen. Live closed captions are available. You can access them by clicking the closed caption button at the bottom of the Zoom video window and then clicking Show Subtitle. You may also customize your caption view by clicking the Stream to Text link posted in the chat. And we are here live streaming today's presentation, which will be archived and available on our website. Of special note, this virtual speaker session is made possible through the generosity of Matthew and Karen Keller, both 2000 graduates of the Eberly College of Science and loyal supporters of the Penn State Alumni Association. The Kellers created this program endowment at the Alumni Association and are sponsoring an annual event that features the Eberly College of Science and its professors and students. The virtual speaker session is the third annual event in the program titled the Keller Family Alumni Experience in the Sciences. Today's guest speaker is Professor Nikki Crowley. She will present her work studying the intersection of neural circuits in the brain, novel neuropeptides, and addiction. She'll discuss how alcohol exposure changes signaling in the brain and how we can engineer therapeutics targets for addictions and mood disorders. In addition, Nikki will present an overview of the unique questions asked and solved by Penn State research and some healthy speculation surrounding the future of emerging technologies and neuroscience. More information on Dr. Crowley can be found in the chat. Thanks for joining us, Nikki, and we look forward to hearing your presentation.
Thanks so much, Paul, and hi, everyone. I'm super excited to be here today. This is actually like a little bit of a special honor for me because I am the child of two Penn State graduates. My parents met here, so talking to the Alumni Association is really special. I'm going to go ahead and share my screen. Can everybody see the right screen? It just looks like my slide deck.
Yes.
This is like the canonical thing that a professor has to say, right? Is can you see my screen? So, all right, I'm going to talk to you guys today about the work that my lab does looking at neuropeptides and how they're kind of unique and special and what we could be doing to manipulate them for things like addiction. I'm also the director of the Neuroscience Institute at Penn State. And I wear some other hats related to neural engineering, but also Alzheimer's and dementia prevention. So in the Q&A, if you have questions that are sort of outside of what I presented on, but really on any domain of neuroscience, I'd be happy to field them and tell you sort of what's going on at Penn State. So my research program in particular is understanding these neuropeptides. And these are really strongly implicated in human diseases, but we really don't understand what they do, in particular what they do in the brain. And I want to understand how neuropeptides sort of uniquely modulate things like neurocircuits to control behavior. So neuropeptides, we're researching them in the prefrontal cortex. And this is probably the brain region that you're most familiar with or you've heard the most about. It's this kind of frontal region of the brain. And it's responsible for all of these behaviors that we think of as sort of executive functioning. So our ability to make decisions, attend to things, remember things, our capacity for language, all of these things are thought to be sort of governed by the prefrontal cortex, as well as sort of the regions that the prefrontal cortex communicates with. But the prefrontal cortex is also really heavily involved in some other things like our capacity for addiction and governing sort of our cognitive decline and things like Alzheimer's disease. So we have sort of this dichotomy of the prefrontal cortex is so important for all of these things that we kind of consider to be uniquely human, but also some of the most sort of stressful things that we can experience. And we think that neuropeptides, at least the ones that we're researching, when they come online, they can sort of tip the valence of how the prefrontal cortex is governing these different systems. So sometimes this can be in a protective fashion. The neuropeptide that I'm going to talk about today, somatostatin, we really think can boost all of these positive attributes, right? Memory, language, our ability to have fulfilling conversations, and that it reduces our likelihood of addiction and cognitive decline. So we think that these neuropeptides are controlling sort of the valence of what the cortex can do. And we're researching this neuropeptide in animal models. The reason we do that is animals, like mice, have a prefrontal cortex that is much like ours. These neuropeptides signal in a similar fashion, but we have a really unique ability to understand what exactly individual cells in the brain are going to be doing when we look at these animal models. So what you're seeing here in this first image in green, all of those little green dots are the neurons, the brain cells in your brain that are going to express the neuropeptide we're interested in. So we can really see where they are and where they're going. We can also see how these neuropeptides interact with other things in a way that we just can't in people. So what you're looking at here in blue, you're seeing all of the somatostatin neurons. And in pink, what you're actually seeing is all of the estrogen receptors in the brain. So the brain receives estrogen as a chemical messenger, and it's going to interact with all of these things. And then we can sort of uniquely understand how these neurons are wiring and firing in real time. So what are neuropeptides doing in the brain in real time, and how are they changing the way the brain signals? And we can do this because the brain is actually electrically active. So all of these neurons are essentially going to communicate both with other parts of your brain and with the rest of your body with an electrical signal. And we can, in animal models, measure these changes in electrical signals to sort of understand what's going on. So this line of work that we're looking at, what we're doing is we're going to take the prefrontal cortex. We want to understand individual neurons in the prefrontal cortex. So what you're looking at in this gray image is the actual brain cells inside of this cortical region that we think is so important for this cognition and language and all of these things. We can poke that individual brain cell with a glass capillary, which then allows us to actually look at this electrical activity of the brain in real time. So we can say, when are these neurons talking? Who are they talking to? And how does it change with things like addiction, or how does it change with exposure to a protective neuropeptide? And what we find is that when we give this peptide somatostatin, when we just want to understand what it does, it ends up quieting some of the circuits in the prefrontal cortex and allowing other circuits to be louder. So what we think is happening is somatostatin as a neuropeptide is kind of dampening the background noise within the cortex, allowing some signals to get through and allowing the brain to sort of communicate specific messages. And we think that this neuropeptide is particularly important for risk taking and sort of communicating the decision to take a risk. And so we can take these electrophysiological principles, understanding when the brain is firing, which circuits are coming on and offline, and we can also look at exactly what they're doing during behavior. So the way that we're going to do this is we have a fluorescent marker that the brain is turning on. And this is really straightforward. We basically can put this color in the brain. It's going to be green. And we can put it in the neurons. And what happens is that every time that neuron is talking to another neuron, that neuron is going to turn a brighter green. And we're going to be able to see the signature of when the neurons are communicating sort of in real time. We can then use a fiber optic probe that's implanted in the mice. They can walk around, they can eat, they're super fine with these probes, and we can pick up that light signature. So we can say, when is this brain region brighter, which is indicating to us that it's communicating more. And this is sort of what that looks like in real time. So what you're seeing in this video is every time you see a green circle kind of flash brighter, that neuron has become active. So what we can do is we can look at how are these somatostatin neurons behaving during very, very relevant tasks. So we're going to take this animal that has this marker of neural activity in these somatostatin neurons, and we want to sort of assess its risk-taking and kind of anxiety-like phenotype. So how are these neurons actually contributing to this? Mice obviously do not have the same anxiety that we do, right? They're not trying to pay their taxes on time. They're not in traffic going to work, but they do have their own sort of ethologically relevant anxiety that they experience. So one way to measure this is using an open platform. And what you're looking at here, it's just a very common assay used to assess the anxiety in rodents. You have this platform that's raised off the ground. They can fall off of it. They don't. But they're really nervous to be out in that open space. And then they have sort of enclosed spaces that are going to be a lot safer for them. And what we can do is we can look at how the animal is behaving in this arena. We can use machine learning to sort of track its movements and the different way that it positions its body. And we can couple that behavior with this neural phenotype that we can measure. So what we found here in E, you're just looking at a heat map of where that animal is choosing to spend time, with the vertical line being that open, risky space, and the horizontal line being that closed, more safe space. And the darker the blue, the more that somatostatin neuropeptide was engaged during the behavior. And what we find is that these somatostatin neurons are really, really active when the mouse is taking risks. So we think that these neurons in the cortex are sort of vital for the decision of kind of like, go ahead and take the risk, right? So it's sort of promoting engaging in this behavior. So risk taking is great. It can be super adaptive, right? We find new friends, we make new social networks, but there is risk taking that's maladaptive. And so one form of that we look at in my lab is binge alcohol consumption. So some alcohol consumption is great. It promotes social behaviors. It makes us happy. But there are high levels of drinking that are not good for us. And so we can look at this in mice, and basically what we found, so here you're looking at the grams per kilogram of alcohol consumed by these mice. All of these mice are legally drunk. They're not allowed to drive a car at this point, right? So they're pitting that 0.08 blood alcohol content that we think about for humans. And what we found is that if we manipulate the somatostatin neurons, if we sort of reduce their activity, we can reduce binge drinking. So we can drive down risk taking by, at least risk taking in terms of binge drinking, by manipulating these neurons. But obviously I've been clear that this is in mice. Since we started doing this work, it's actually been thematically replicated in humans, which is really exciting for us. So this is work looking at young men who drink and looking at their sort of fMRI change, the change in their brain activity that happens when they're drinking. And the gist of what this group found was that the more somatostatin that a human individual has, they are also protected against the sort of the risk that comes to the brain from binge drinking. So this was really exciting for us to see our sort of preclinical work translated to actually into the human clinic. And then I'm gonna have a couple QR codes for you in case you want to read more about certain things. So if you're interested in understanding sort of the role that we think stress and alcohol are playing in brain health, I recently talked to Newsweek about this. So this is a great article on, you know, if you're going to be binge drinking, maybe don't do it when you're super stressed. So we're really interested in how these neuropeptides promote risk taking, and that we can manipulate this system to reduce binge drinking, but we can't always do that, right? So a lot of us maybe drank a lot more when we were younger, and now we're nervous about it. We don't drink so much today as we get older. So we're really interested in understanding what happens if the alcohol exposure came first. How does this muddy the water? So what happens to this kind of picture of what these peptides can do in the prefrontal cortex if we have life events like binge drinking that have already happened? Can the prefrontal cortex still function properly, and do these neuropeptides still function properly? And if not, what can we do about it? This is also lends itself really, really well to animal models because it's very fast. So if we think about the time it would take to understand this in people who might have binge drank when they were preteens all the way through college, this is a very short window of time when we're talking about mice. So they're reaching their adulthood in only a couple of months. So we can give them alcohol in a binge drinking fashion just while they're young and sort of look at the long-term ramifications on their brain health. They will, again, drink a lot of alcohol. So here you're looking at females in red and males in purple, and we're looking at how much alcohol they choose to drink during this adolescent period. And something you might note, you can just tell by eye that the females are drinking a lot more than the males. This is really interesting. We are sort of seeing this in our society as well, and I'm happy to answer questions on it at the end. But we find this to be a really important question because almost 1 million adolescents in the United States are meeting the threshold for an alcohol use disorder. This is how many are diagnosed. So you can imagine that there are many, many young individuals who are binge drinking who are not getting a diagnosis. So we're really interested in understanding what can we do for these brains that have undergone so much alcohol exposure. And sort of broadly, what we had found is that when we layer on adolescent binge drinking and then we look at the brain later in adulthood, this somatostatin system that I just talked about that promotes risk taking is way off course from what it's supposed to be. It never sort of appears to go back to normal. So if we take this healthy functioning prefrontal cortex in this box, we've really disrupted global communication with adolescent binge drinking. And there's really interesting, important behavioral ramifications for that too. So here I'm going to show you males and females in a different color. The assay is still that sort of elevated plus maze we talked about, but just think about the two arrows, which way is going to be more anxious and which is going to be less anxious. And basically what we found is that adolescent binge drinking is making the adult male mice less anxious, but the female mice are becoming more anxious. And this is dependent on the amount of alcohol that they drink. I don't want to, overthink what we mean by more or less anxious, because again, some form of anxiety is really adaptive, right? It's great not to walk into a highway. It's appropriate to be scared of that. But obviously having zero anxiety is going to cause you to take a lot of risks that maybe you shouldn't. So we're really interested in understanding now that, and I want to note here, we're looking months and months after alcohol exposure. These animals are not still drunk. They haven't had alcohol in a long time. So what we're seeing is that binge drinking during adolescence is leading to this permanent change in how the brain and how the prefrontal cortex and these neurons is going to modulate anxiety. And then here you can read my thoughts. I recently did a piece through the conversation on these sex differences that we see. Why are men and women drinking differently, and how should we be handling that? So just to kind of go over like the big picture stuff from the work my lab does, we know that somatostatin neurons in the prefrontal cortex appear to promote risk taking. They seem to be active. They're highly communicating with other neurons when engaged in a risky task. And that this appears to happen due to the peptide kind of dampening the noise of certain neurons. We can manipulate the somatostatin neurons to reduce harmful risk taking, right? So we can reduce the amount that an individual in an animal model is binge drinking when we manipulate these neurons that promote risk. So we have a mechanism to drive down binge drinking. However, early life events are disrupting the system, right? We can't go back and change how much we drank when we were younger or the stupid decisions we made. And this is a big problem, right? So we've already changed the brain. What do we do going forward to fix it? So this is terrible, right? We have plans to help though. And this is a really interesting technology forward project that we're working on at Penn State, which is the idea that we could non-invasively modulate these circuits in the adult brain. And we're doing this through ultrasound neuromodulation. So this is a mouse that's wearing a transparent ultrasound on its skull, allowing us to stimulate through the skull and change neural activity. If you had told me 10 years ago I was going to work on this, I would have told you that's science fiction, but it's amazing. And it's being done in human clinical trials too, right? So not everybody's going to want an electrode implanted in their brain to sort of modulate their behavior, but if we could modulate signaling externally, that would be a great therapeutic. And so we're exploring whether we can do this to restore the function of neurons sort of after the drinking event has occurred. Ultrasound is really, really safe. We're talking about the same technology that we use on pregnant women. It's FDA approved for a lot of things. So to us, this is a really exciting step forward in what technology we can do. And we're doing this with a huge team across not only the US, but the world. So we have clinical collaborators who are doing this at UVA, here at Penn State, obviously, the University of Wisconsin, looking at how we can do this in people. And then we have research collaborators at just some amazing institutions that are really excited about the technology that is in development at Penn State and want to be a part of it. So I want to take a minute to think about what I think is like the next interesting things to be answered. So the National Institute of Health is really focused on the Brain Initiative, which is to revolutionize our understanding of the human brain. And sort of the next evolution of research into the human mind. So that's a big question, right? That's a wide open field, is what are the important things we need to solve going forward? I'm going to show you these two pictures that I want, that are going to make my point. So this is my son. He's turning 9 on Sunday at the Penn State Arboretum. It's a favorite place to go. And this is my daughter. She just turned 4, 2 weeks ago. She wants to be a brain scientist. She's so excited. And so I think about, we have lots of big questions in neuroscience we want to ask. I think we're going to solve all the big things. I think we're going to solve Alzheimer's disease. We're going to solve addiction. We're going to solve post-traumatic stress disorder. All of these things I think are scientifically very much in our grasp, in the next decade. And when I look at these two kids and sort of what's going on and what we want to solve, I think about this term that I call physiological flourishing. So what does the healthiest brain look like? Can we turn back the clock on the brain back to that healthiest state? You know, anybody who's been around kids, their brains are so plastic, their capacity for memory and language is so high, and they think such interesting things and wonder. And so I think Penn State is uniquely poised to sort of understand this question of what is physiological flourishing, and can we have that brain in our own bodies for as long as possible? So with that, I want to thank the people who actually get to do all of the really cool science. I have a lot of postdocs and graduate students in the lab. We probably have, 10 to 15 undergraduates working with us across all departments on campus who are really passionate about neuroscience. We collaborate really interdisciplinary across all of the colleges at Penn State to do some of this work. And then I just want to note, we have a lot of neuroscience at Penn State. So we have the Institute, which is comprised of over 65 faculty. We have 20 students doing PhDs in neuroscience, doing their doctorate in neuroscience, dozens more that are focusing in other areas that interface with neuroscience. And then we have centers within our institute that are specializing in really different domains. So neural engineering, neurotechnology, and mental health, right? How do we make the best technology for some of these disorders? What is the neural basis of cognition, right? How do we have consciousness and thought? And also, for example, the Center for Molecular Investigations of Neurological Disorders is really focused on solving the diseased brain. Yeah, so with that, I'm happy to have Paul modulate Q&A.
Excellent. If you have any questions for Dr. Crowley, you can drop them in the Q&A tab on the bottom of the screen. Nikki, would you mind stop sharing?
Yeah, got it.
Okay, and then hold on, I'm just trying to get the screens so that people can see us. Let's see here. All right, there we go. So so fascinating. I actually heard your presentation during the lecture series that happens over at Huck on a Saturday afternoon.
Yeah.
I happened to catch that. It's fascinating research, especially with the prevalence of binge drinking in a college town, right? We were talking about life in a college town before we got on here, and for better or worse, this binge drinking is part of college town culture.
Yeah.
So we have a couple questions that have come in, and let me get to them. First one from Jeff. When you're looking for the cure for most diseases that you research, right? Is it a matter of time or money at arriving to the breakthrough?
That's a great question. Yeah.
Barrier?
So what's the biggest barrier to sort of groundbreaking research? I'm going to answer it, and I'm going to tell you how I think Penn State actually overcomes that. I think the biggest barrier is interdisciplinarity. And what I mean by that is you can take a lab like mine who can find a target, and we can say, this is the neuron, this is the neuropeptide that you need to look at. But that's sort of where our expertise ends. And we need other people to come online and say, I'm going to make the druggable version of this peptide that we can get across the blood-brain barrier and that doesn't have side effects and all of these things. And you need other people who say, well, I'm a prevention scientist, and I'm going to come in and I'm going to figure out what is the earliest time point we can identify this problem. This is really hard to do at med schools, and sort of some of these more, a lot of the Ivy League universities, people end up being siloed in one domain, and they don't reach out and they do that kind of work because they work from a perspective of competition and not collaboration. At Penn State, however, I think that if you are a neuroscientist working at Penn State, you believe in the land-grant mission of Penn State and you believe in collaboration. And I always describe neuroscience at Penn State as a rising tide lifts all boats, right? We want to see interdisciplinary science succeed, and a success in a lab in agriculture is a success for all of us. So I think we're really building a culture where we're going to do some of these things at a higher level than you might see at a university where everybody thinks about themselves. IM has never been the Penn State mentality, and it's not for research either. So I'm really excited to see us overcome this hurdle of interdisciplinarity is really necessary for brain science.
It's interesting that you point out where the research is actually occurring. one of the interesting talking points here at Penn State is there's actually more federally funded medical research going on at University Park than actually happens at the hospital. And I think it's for the reasons that you've just talked about.
Yeah, I mean, we believe in working together. I'm constantly knocking on doors, and except for right now, I've got a do not knock note on my door, but usually my door is open. And, I've had people reach out to me and they say, hey, we're looking at something in the College of Ag related to egg production in our species. And can we understand how alcohol impacts that or what the interactions will be? I don't know how to emphasize enough that you don't find this everywhere. That the idea that you were supposed to do interdisciplinary work and cross boundaries is where discovery happens. And it is not only happening at Penn State, it is an expectation that is how you run your lab and you execute your research, is that you need to push boundaries.
So a question coming in from Sean. He wants to know, how can you rewire the signaling of an alcohol-addicted brain to not require alcohol to experience satisfaction over time?
That's a great question. Okay, so the question is sort of how do you, if the alcoholic brain is experiencing a lot of satisfaction from alcohol, what do you do for it? So what we actually find in individuals diagnosed with alcohol use disorder is they don't get a lot of satisfaction from alcohol. So sort of over time, what's going to happen is that you will start taking alcohol just to not feel crummy, right? We probably all know what a one-off hangover feels like, and it's terrible. But the more that you have those cycles of alcohol consumption and then anxiety and sort of, we call it the hangxiety, right? The hangover anxiety, the more you're going to feel that anxiety at any given time, the more you're just going to have sleep issues at any given time. And so a lot of times what happens with alcohol use disorder is you're not drinking to feel great anymore. You're drinking to not feel bad. So it really is this interesting rewiring of how pleasure and stress interact in our brain. And that's actually what I wrote about for the conversation, if people want to check it out, is the idea that we sort of have a shift up from something being enjoyable to just trying to feel baseline again, right? And so that's what we have to think about in terms of rewiring the alcoholic brain, is how do we sort of reset the stress threshold and reset the anxiety threshold so that alcohol could then be rewarding on occasion again, right? And so in terms of actually doing that, that's a great question, because now we're talking about interacting with other mental health conditions, right? So we want to get you back to a baseline of feeling good, a stable mood, but without increasing your propensity to do other things that are bad for you too. So that is a really, really active area of research. And that's kind of what I would say is our main focus, is how do we get that brain back to a neutral state?
David has a question. What are the implications for substance use disorder?
Yeah, so for us, we have sort of this, I'm going to call it two-pronged approach across the lifespan, which is if the brain is already addicted, what can we do? We think that somatostatin as a therapeutic is going to be a really good target. So the question is, can we pharmacologically administer this? Can you take it at your doctor's office to dampen that binge drinking? We know it's effective. So is the neuropeptide itself a good target? We think it is, and we're going to move forward with that. The other sort of kind of part of the lifespan that we look at is, can we identify the brain that is most likely to develop addiction and sort of target our treatments towards them? So lots of people drink alcohol. A small percentage of them are going to develop a substance use disorder. So we're really interested in what is the earliest signature that alcohol is changing your brain differently than someone else's brain. And can we step in really, really early? That would be great. If you knew which high school students were going to a party and were going to choose to binge drink and then are going to become a lifelong alcoholic, you could step in really early, but not everybody's going to need you to step in, right? So identifying the earliest intervention point, or sort of the earliest signature of brain change, is also how we're thinking about this for substance use disorder.
So I realize this presentation is about kind of alcohol and alcohol addiction, but I know there's numerous other kinds of addiction out there, right?
Yeah.
Is there an addiction center in our brain, or is it different parts of our brain that are triggered by that trigger different kinds of addictions?
Yeah, great question. So I'm gonna give this a multifaceted answer that's my opinion. Okay, so the first is that for things related to brain disorders, we want them to have joints. Our way of thinking about things is to treat it as a discrete thing. So like if I break this part of my arm, we know that I didn't break this part of my arm because they're separated by a joint. That tends to not be how the brain works, right? There's a lot of overlap and sort of color blending of what is addiction, what is OCD, what is anxiety, what is mental health. So all of these different things, sort of all addictions are going to have sort of an overlapping neurocircuitry. We don't know the sort of entire pathway of what addiction's doing in the brain, or hopefully we would have solved addiction by now. But it does interact a lot with sort of our movement centers. And this is so interesting if we think about alcohol consumption, smoking cigarettes, gambling as sort of repetitive behaviors, right? These are things that we physically do with our body over and over again. And some of those things, like riding a bike, become muscle memory, and that's awesome, right? We don't want to think about riding a bike every time we ride a bike. But we do want to think about drinking every time we drink, right? We don't want the choice to drink alcohol to be muscle memory like riding a bike. So we do think that some of the circuits that are involved are going to be the circuits that govern movement disorder. And then we have this sort of dichotomy happening of the circuits in your brain that govern reward and the circuits of your brain that govern anxiety. And the prefrontal cortex is going to control both of those systems. And so what we think is happening is the prefrontal cortex is kind of shifting its ability to control reward towards promoting anxiety behaviors. And so we've got to shift that balance back to be balanced. And it is likely that all addictions sort of have overlapping circuits that they're using.
So a couple questions coming in, maybe off topic, but I think within your research.
Yeah, that's cool.
Interested in, Lori has a question about the use of hallucinogenics in the treating of mental health issues.
Yeah, okay, hallucinogenics like LSD, ketamine, psilocybin, these are so interesting because I will say sort of broadly, when we think about things that influence the brain, we often can know that they influence the brain and that they're effective before we understand how they work, right? There's thousands of years of traditional medicine that can support that these things can be good. So it's a really hot topic right now, right? Can we take hallucinogenics to influence brain health? The literature is clear that some of these things are going to have positive outcomes when coupled with appropriate therapy, right? There is a big, big difference between taking ketamine for depression with your doctor and taking ketamine at a bar. Don't advocate for the latter. Talk to your doctor about the former. I think all of them are really exciting. The data coming out for ketamine, for mood disorders, for psilocybin, for LSD, it's really strong. I think that I'm happy to see NIH invest in these things. The other thing that's so interesting about some of these drugs is the positive outlook that you can get from them appears to last for a really long time. So a lot of the original work that came out with like psilocybin or mushrooms was related to terminal cancer patients. And so they were able to see that terminal cancer patients had an increased positive outlook for a very, very long time after a single exposure. So I think this is a really hot area. I'm really excited about it. I'm super supportive of the work that's being done there. Don't do it in your garage though, but I think it's really promising.
I think that's good advice for most science, right? At
Yeah.
least science that involves humans as the test cases, right? Let's not do it in our garage.
Yeah, wait it out a little bit longer.
Exactly. So you've mentioned some of your other research on dementia and Alzheimer's. Are there valid indicators of likely treatments or medications to stop, reverse, and even reverse dementia or early Alzheimer's disease?
Yeah. Okay. So sort of what is our understanding of Alzheimer's right now? Prevention, intervention. I don't know how effective the therapeutics are going to be to reverse Alzheimer's disease. And that is because, we have to consider for some of these things that the neurons don't exist anymore, right? If the brain has been damaged, sometimes there's nothing we can do to get that back. So we have at Penn State the Neurosciences and Dementia Prevention Consortium, and I'm the associate director. I oversee neuroscience for that. And sort of the mentality that we are taking goes back to this idea of prevention, of what's the earliest that we can do to identify that somebody might be on the course for Alzheimer's before the brain is damaged, before there's amyloid beta or tau tangles or any of these things? So if we can find a biomarker that signifies that that brain is going down that path before it's been disrupted, that's what we really need, right? And there are really promising targets, really exciting things that people are thinking about there. So one of them is this idea of inflammation in the brain really driving Alzheimer's disease. So have there been events that have driven sort of increased inflammation? Just like we think about inflammation not always being good anywhere else in your body, it's not great for your brain either. So the Neurosciences and Dementia Prevention Consortium, we're really focusing on finding these biological signatures that happen early before any cognitive decline. And we're doing this in really interesting ways, looking at both, these rapid mouse models and humans, because it's really hard to have fast therapies for aging disorders in humans, right? If you've got to wait 70 years for the data, it's just going to be slow. And we can look at an adult mouse in about 2 years. So we're looking at sort of what are the environmental factors that are leading to inflammation that's causing cognitive decline, and can we prevent it early? In terms of other things that I think are interesting for dementia, somatostatin, the target we look at, is super, super interesting. I've already explained that I kind of feel like it's very protective. It promotes sort of risk-taking, which is a form of flexible thinking. So there are FDA-approved somatostatin drugs that are actually used off-label for cognitive decline. And what they find is that individuals who have diagnosed Alzheimer's, if you give them somatostatin, their working memory improves. They seem to do better conversationally with their families. So we have a line of work where we're diving into trying to understand what the mechanism of that is, and would it be good for all dementia or just some? So really hard to do, interventions in a patient when they already have dementia, because it depends on sort of how intact their brain still is. And I think that the most valuable work coming from a neuroscience side is going to be sort of thinking earlier in the progress of disease.
So a couple questions. I'm not an expert in this field, but there may be some connectivity or overlap on these. So let me ask kind of both of them. Cynthia wants to know if there are kind of what the implications for eating disorders are, and if your work is looking at those. And then Stephen's asking about evidence linking the biome of the gut to brain cognitivity.
Yeah, okay. I'm going to answer the first one first and then remind me of the second one. Okay. The link between eating disorders and binge drinking is really, really clear. They're both kind of consummatory behaviors that we do to excess. What we are also seeing, which is so exciting, is, obviously lots of people in America are on GLP-1 targeting drugs for obesity right now. And what has happened is that just by nature of how many people are on those drugs, a lot of alcoholics are also on them, even if they're not taking those drugs to treat their alcohol use. They're taking it to manage their relationship with food, and it's decreasing their binge drinking. So we are seeing alcohol rates go down a lot when people are taking some of these drugs that have been made for obesity. It's super interesting. There are important limitations to that and things that people need to be careful about. For instance, there's a lot in the news about like the rebound effect that can happen with Ozempic and these drugs, that when you stop taking them, you'll start eating a lot again. We are also seeing that with alcohol consumption, and someone will not drink the whole time they're on their drugs, but then when they stop taking Ozempic, they'll try to drink a lot of alcohol again. And that in some ways is much riskier, ramping up alcohol really, really fast, than just, eating a lot again. From a safety standpoint, right? If you haven't been drinking for 6 months, you can't have 10 beers. You're going to be really sick from it.
Especially if you've experienced significant weight loss also in that period.
And, there's this layer of tolerance, right? The more that you drink, the more you can drink. But if you haven't been drinking, you might go back to your former consumption levels and you weigh less, and it's going to hit you really hard. But that being said, it's super exciting that these drugs are working. I have been following the news a little bit about concerns about side effects related to mood. And this is what's so difficult is, you as an individual, me as an individual, I don't want to be addicted to anything, but I do want to experience reinforcing things, right? I want to have joy from my family and from exercise and from my work. So it's really about trying to dial that in to say, you know, it's going to reduce my drinking, but it's not going to reduce how much I enjoy my life. Okay, second question was microbiome.
The link, yeah, the linkage between kind of the biome in the gut and brain cognitivity.
Yeah, I can tell you that Penn State is going to be a leader in this space because I'm directing the Neuroscience Institute and we have a world famous microbiome center here, and we're already talking about ways to tie these projects together. Alcohol is absolutely going to influence your gut health. I'm a big believer in, like, we have a gut instinct, right? We have things that go in our torso, and we know that they're influencing our brain, right? We can feel sick when we're anxious. So there's definitely ties between the signaling in the periphery and the brain. The microbiome in particular has not been well investigated with alcohol or with brain health in general. And I'll tell you why. This goes all the way back to my first point, that you've got to promote interdisciplinary science. You are not going to find someone who's an expert in the microbiome and an expert in the brain. It's too many things to be an expert in. But we have both here at Penn State, and so we're going to do both, right? Penn State believes in promoting interdisciplinary science. So there's not a lot on this question, microbiome and addiction or microbiome and brain in general. It's growing, and I think you'll see more of it come out in the next couple of years.
So last question that we have is, how can a physician who is fascinated by neurological disorders that overlap with anxiety participate in work to advance this science at Penn State?
You can email me. I am really passionate about making sure that the work at Penn State is being grounded in actually helping human health and sort of helping the therapeutic process for patients. You know, I always tell my lab, we're not in it to be expert mouse neuroanatomists. We want to solve the brain to help humans and to help healthy living and enjoyment of life. And so I showed on one of my previous slides that we have clinical oversight, people who are just sort of volunteering their time to make sure that the way we're thinking about experiments and designing experiments where feasible is sort of mimicking the human condition. So for all of our models of binge drinking, they're really, really well grounded in the way that humans binge drink, sort of the pattern and time in which we do. So we're sort of always welcoming clinicians who want to provide feedback or tell us where our models might miss the mark or where we're not thinking about something, right? We absolutely know as basic neuroscientists that we can't model everything that's going on for people, but we want to be doing the best job that we can.
Excellent. If people are looking for more information on the research that you're doing, where can they go to find that?
So you can go to the Penn State Neuroscience Institute. That will link to all of the labs in neuroscience at Penn State. So that if you go to the Huck Institute, it's huck.psu.edu. That'll get you there. My lab in particular, we are Crowley-lab.org. And that'll tell you more about the work we're doing, who's in our lab, sort of, the PhD students, the undergrads, where they're all coming from. So go to the Huck to find out about all neuroscience and go to crowleylab.org to find out about mine.
Well, that's all the time we have for today. I'd like to thank Dr. Nikki Crowley for joining us. And I also want to thank all of you who tuned in to today's virtual speaker session. As a reminder, you can view a few listing of all of our events online and in person at alumni.psu.edu/events. Thanks again, and for all you do for the university, for the glory, and for the future. We are
Penn State.